Welcome back, everybody. This is part 3B of Unit 1. This, in fact, is going to be our last part. We are going to be tackling Chapter 8. Chapter 8 is going to be very heavily based on definition terms. We're going to go ahead and start exploring the structure of the chromosome. We'll take a look at the centromere. We'll take a look at some abnormalities. So for instance, if you have like a deletion or an inversion on your chromosome, what does that mean? How can it affect the organism? And then towards the end of the chapter, we're also going to take a look at what happens if you start altering either a certain amount of chromosomes or just a complete set of chromosomes that an organism can inherit. But before we get into all that fun, let's take a look at the centromere. Now, the centromere is going to be the attachment point that allows for the two sister chromatids to physically connect to each other. So here they are right here, highlighted in the yellow structure. And we see that the position of the centromere can be varying depending on which chromosome we're looking at. If we see that the centromere is pretty close towards the middle of the entire chromosome, we call that metacentric. And as we're looking at this illustration, what you're also going to notice is that the chromosome has been labeled either P or Q. And those two letters are going to indicate either the P, which stands for petite, which is the short arm of the chromosome, and Q is going to stand for the long arm of the chromosome. Both of these letters are used for locational terminology. So for instance, if you're reading an article and it says to you, this particular disease can be investigated or can be located on chromosome 2 by the Q, that means that it's located on the second chromosome on the long arm. So P means the short arm, Q means the long arm of the chromosome. If we see that the centromere is slightly off the middle, then we call it submetacentric. So we can go ahead and take a look at our second illustration right here. And you can see that the centromere is slightly off. Obviously, there will also be a scenario where we'll have more of a off-center location where it will favor one arm over the other. If we see that happening with our third example, for instance, that is then called an acrocentric centromere. And the last one is the most extreme. This is when we have a centromere that's located all the way at the end of a chromosome, and this is then called telecentric. Okay, so we have metacentric in the middle, submetacentric a little bit off, acrocentric favoring one side over the other, and then telecentric actually towards the end of your chromosomes. A lot of what we know about the physical appearance of the chromosome and the location of the centromere is due to the fact that we have a Kamasi stain or a G banding pattern that can be utilized so that you can visualize your chromosomes under the microscope. And in fact, what you see here is an illustration of the G bands that you would come across if you were to stain our 23 pairs of chromosomes. Now, when you look at the chromosomes, you're going to notice that the left side of the chromosome is not equal to that of the right when it comes to the banding patterns, and I'm talking about the black areas, and that is because it's showing you the chromosomes at two different stages of the cell cycle. On your left-hand side, on the left-hand side of the chromosome, you see the banding pattern during metaphase, and on the right-hand side, you see the banding pattern during pro-metaphase, so when the chromatids are a little bit slightly more extended. And it basically shows you the dark areas are going to be areas that are tightly compacted and that are able to absorb the staining pattern. So just wanted to kind of show you this so you can take a look at the overall formation of the band patterns. Now, the bulk of the chapter is going to take a look at mutations that can alter our chromosome structures. And these mutations basically will fall in four categories. We can have what we call a deletion, which is also called a deficiency, but most books and research papers will call it deletion, so we'll kind of stick to that. You can have a duplication. you can have an inversion, and you can have a translocation. And obviously you could see that each of them come with a short little definition, but I think it's better for us to kind of visualize it, and then what we'll do is we'll take a few minutes out and we'll discuss each of them individually, and we'll take a look at which one is more detrimental and which one is more forgiving when it comes to overall expression of the genes. Okay, so let's start with this illustration right here. And what we're seeing are our 4 categories. So on the top part for A, follow the little red arrows, what we see happening is that if a deletion occurs, as you might indicate from the name, that means that we're going to delete a certain section of the chromosome. So if this is our wild type, what we see happening in our mutated version is that if the area between the 2 red arrows is going to be deleted. So if you come over here to the mutated version, you're going to notice that there's no area marked 2 between the 3 and the 1. So you are deleting a section of your chromosome, you are shortening it, and with that you're also deleting maybe some essential genes. Another example of a mutation would be a duplication, and it's all in the name once again. A duplication means that we double up or we increase the amount of genomic information in a certain section of the chromosome. So once again, if you go ahead and take a look at your wild type on the left, see the 2 little red arrows. What we see happening in our example for our mutation for duplication is that the area between the 3 and the 2 has been doubled, thereby allowing for more genomic information to be inserted into the chromosome, and now you have more genes, more DNA than normal. An inversion is when we are going to flip a certain section on the chromosome. The good news with an inversion is that you have the same amount of genes. The bad news is that the genes are just in an alternate order. So in this example right here, once again on our wild type on the left, we see that we're going to take a look at the area between the 3 and the 2, follow the red arrows, and what we see happening in our mutated one is that we still have all the same genes, with the exception that now the order of the 3 and the 2 has been flipped. So this is your inversion. So in both the deletion and the duplication, you are altering the total amount of genes that you have. Whereas in an inversion, you keep the same number of genes, but you simply flip the order that they're in. Then our last example that we're going to be discussing is going to be called translocation. And what you want to keep in mind with translocation is that we're going to be altering segments of chromosomes from non-homologous pairing. So chromosomes that are not naturally meant to swap material from each other. So for instance, right over here, you can see you have your 2 chromosomes lined up. One is going to be a very long one is a nice little short one. And what we see happening is that with translocation, that means we're going to swap genetic material between non-homologous chromosomes, and the swap can occur 2 ways. You can have what we call a simple translocation or a reciprocal translocation. In a simple translocation, we see that the material is removed from one and donated to the other. So if you come over here to the mutant, notice how this is all gone, and this particular area of genes has now been moved over to your non-homologous chromosome, right over here. In a reciprocal translocation, that means that both of the non-homologous chromosomes are going to swap material with each other. So if you come over here now to the mutated one, you're going to notice that both the tall as well as the short chromosomes have received reciprocal information from each other. The good news for the reciprocal translocation is that, once again, the total number of genes is kept the same. However, one thing that you're going to encounter is that when it's time to produce your gametes, there might be some issues with separating the genes in a proper haploid formation. In a simple translocation, what you're doing is you're altering the length of both of those chromosomes dramatically. One of them will shorten while the other one will lengthen. And also keep in mind that with a simple translocation, you only move the material from one chromosome to the next, so it just attaches to the second one, whereas in a reciprocal translocation, you have two separate chromosomes that swap materials with each other. Once again, pointing out that these are non-homologous pairing. Alrighty, so let's kind of like dive into a little bit more each of our examples. So our deletion, our duplication, our inversion, and our translocation. Let's take a look at some more examples, and maybe we can come across some disease models as we chit-chat about it as well. So let's start off with the deletion. Now, the deletion can be detrimental or it can have no effect. All of it will depend on exactly which chromosome is being affected. Are you affecting an area that's labeled junk DNA where you have no transcription or translation that's occurring? Is it an area that is where you have your vital or your non-essential genes? It will all depend on the overall location where this is occurring. The size of the deletion is also very important. The larger the deletion is, the more chance you have that it could be detrimental. One thing you want to think about is that the bulk of our DNA falls under non-genomic DNA. We basically only use about 2 to 3% of our DNA for our actual genes for transcription and translation. So just because you have a mutation doesn't mean that you'll automatically have a side effect or a detrimental or even a beneficial side product from it. It all depends on where this particular mutation occurs. A lot of times the mutations will occur in areas of the DNA that aren't utilized, so you wouldn't have any idea that that mutation is even there. The other thing that I want to point out on this slide is that when we're looking at our deletions, oftentimes it's further subdivided into what we call a terminal or an interstitial deletion. A terminal deletion means that you basically break off the end part of your chromosome in its abnormality or in its mutation. So for instance, if you come down over here and take a look at A, this is an example of terminal deletion where the end part of the chromosome, so genes 3 and 4, is broken off, and now the mutated version of the chromosome is shorter due to losing its end piece. If you see that the break occurs within the middle or center of the chromosome, then we like to call that an interstitial deletion. So here's our example of interstitial deletion, the illustration on your right-hand side. And now what we see happening is that this piece right here between gene 3 and 2 is going to be chopped off from the wild type, producing a mutant chromosome that is shorter than normal. And because the deletion came from the middle part of the chromosome, we call that an interstitial deletion. An example of a clinical application for your deletion we can find on our next slide. It is called Lejeune or Cridachat syndrome in humans. What we see happening is this is a dominant disorder where we have a deletion on the short arm of chromosome 5. So here on our illustration, you can see our wild type. And then here's our mutated version where we're missing a little piece of the short arm, and the deletion of that information, and by the way, that would be an example of a terminal deletion because it's at the end. The deletion of that particular piece of information causes the individual to develop some mental deficiency. We also usually see that from a phenotypical aspect, they have some very unique facial characteristics that develop. But probably one of the main characteristics of just genetic disorder is that as a baby, they have a screeching cat-like scream, and that is basically what the syndrome, the symptoms, or sorry, the disease is named for. In duplications, now we're going to be adding genes to our overall genome, and very similar to when we were discussing our deletions, it's all going to depend on where it is happening and how much are you actually duplicating. Usually what we see happening is that if you compare a duplication to a deletion, your duplication tends to have more likely to have a phenotypical effect just because you're adding more material, and it tends to affect a larger number of overall genes than when you compare it to a deletion. Usually what we do happening is that the chromosome duplication tends to align or tend to occur when you have something called non-allelic homologous recombination, or when you're looking at a chromosome that has a lot of repetitive sequences by it. And when that chromosome tends to replicate because of the repetition, chances are that a mistake will be made that can cause a duplication to arise from it. So here's my little example. Let's go ahead and take a look at what I was mentioning to you before for the duplication, that it can be caused by a non-allelic homologous recombination. So what we're looking at over here are our two chromosomes. These are homologous chromosomes. And what we see happening is when they align up during metaphase, they will basically go ahead and they will swap materials from a non-allelic standpoint. So what do I mean with that? So even though these are homologous, what we see happening is that the repetitive sequences within them is going to cause the swapping to occur. So instead of B and B swapping together, C and C swapping together, we see that the B and the C will go ahead and start intermingling together, thereby causing you to have a misaligned crossover event to occur. So as the end, what you see happening is that some of the chromosomes will have duplications, where the material is in excess, and others will have deletions, where the material is missing and has been shortened. And like I said to you before, this tends to really happen in the repetitive sequences. It's almost like making a spelling mistake, so when you ask someone, for instance, to spell a word like Mississippi, sometimes they'll get confused as to how many S's and how many P's, in what order are you putting it. It turns out that we also, in our genomic information, make mistakes when there are repetitive sequences. And this can actually increases our chances of forming duplications, primarily between the non-allelic region in homologous chromosomes. One good thing that can come out of a duplication, though, is that it can start forming what we call a gene family. So oftentimes when you do duplication, you basically literally just photocopy a particular section of genes, and these genes will have a high amount of similarities. Over time, these two genes can start mutating independently from each other, and they can start each coming up with their individual function, slightly altered from their original version. So what we see happening, therefore, is that these duplications then become part of a gene family. So they're very similar in function, but each of them can have their own specialty. If we see that these genes, if it's two or more of them, are derived from a common ancestor, we like to call them homologous, obviously. And if they're found in the single species, we also like to call them paralogs. So homologous just means that they have the same ancestry, and paralogs means that they're coming from a single species when we're looking at their originality or their start. A really good example of a gene family that we utilize throughout the day is basically when we take a look at hemoglobin and myoglobin and all the genes that regulate our globulin formation. Let's go ahead and switch over to the next slide, and I'll kind of chit-chat and I'll show you what I mean with all that. So here we have our regular piece of DNA on top of the illustration, and as you can see, we have a gene that's been highlighted. And let's say that you have an abnormal duplication, and for one way or another, we duplicate this particular gene. So here you can see on our second strand, we have one version of the gene, and we have the second version of the gene. Perfect example of gene duplication. So you have more than you normally would have. If we see that as the organism continues to replicate, these two genes also replicate by themselves, and we see that over time the genes start accumulating mutations independently, what we then see happening is that the genes can often become more specialized in their function, and they can then start becoming part of a gene family. You can also see that on the illustration that they've labeled them as paralogs and homologous genes. Paralogs because they're within the same species, homologous because they are coming from a common ancestor. So the example that I was chit-chatting to you about is the fact that when we take a look at oxygen binding to the red blood cells, we see that the gene that will go ahead and stipulate the globulins can either be, for instance, myoglobulin or hemoglobulin. Myoglobulin is more specific for our muscle cells. Hemoglobulin is what we encounter when we take a look at our red blood cells. Both of them will function in tracking and attaching oxygen to the red blood cell. However, they will do it slightly different based on the composition of the overall cells that they're serving. So we see that the two genes, which are basically the end product of a mutation, have accumulated their own specialized function along the way and are now part of a gene family. Alrighty, now another example of a mutation that you might encounter when we take a look at our chromosomes are inversions. So inversions literally means that we just kind of flip the orientation, the order of a particular gene on the chromosome, and it's very important to point out that unlike deletions and duplication, the total amount of genetic information is staying the same. And because the total amount of genomic information stays the same, what we see happening is that the great majority of inversions have absolutely no phenotypical consequences because you still have the DNA that is required or the genes that are required to produce the protein that the body is in need of. Now, if the inversion happens within the centromere region, we call it a pericentric inversion. If we see that the centromere is nowhere near the flipping of the orientation, we call it a pericentric inversion. So we have to mention the centromere again. Let's take a look at this from an illustrative standpoint. Alrighty, so here is our wild type chromosome on top, and here's our little centromere. And what we see happening is that if the inverted region occurs and it involves the centromere, then we're going to go ahead and call this a pericentric inversion. If the inverted region is occurring outside of the centromere, as you can see right here, does not involve it at all, then we call it a pericentric inversion. Now, I don't want to say that there's absolutely no phenotypical consequences. There are some rare cases where you can have some alterations. Most of that are going to include what we call a breakpoint or a position effect. So I definitely want to kind of chit-chat to you about that on our next slide. Oh, I'm sorry, I forgot about my little blue boxes. So right over here, you can see the centromere lies within the inverted region for pericentric, and outside the inverted region for the paracentric, peri and para. Alrighty, so if we see that the inversion can alter the phenotype of an individual, then oftentimes what we see is that you have a break that occurs within the vital gene section. These are called breakpoints. Or that the gene is repositioned in a way that it will completely affect the phenotypical expression of that gene. That is then what we like to call a position effect. And oftentimes what we see happening is that about 2% of the human population will have some inversions that you can see through the use of a simple G staining and a light microscope. The majority of these people will absolutely be normal, quote unquote, from a phenotypical aspect. However, some of them, when it comes to producing offsprings, their gametes tend to have some issues which can lead to some abnormalities. Alrighty, so let's take a look now at what we call inversion heterozygotes. Inversion heterozygotes is when you come across an individual that has one copy of a completely normal wild-type chromosome and one copy that is inverted. So one copy, the normal order, A, B, C, D, E. On the other side, you're going to do inverted. So we're going to go the other way. Now, what ends up happening here is that the organism, because they still have the same amount of total DNA, the individuals tend to be phenotypically absolutely normal. There tends to become an issue when it's time to produce the gametes, because remember, when you produce your gametes, what you need to do is you need to go ahead and do crossing over, and in order for crossing over to be successful, we're going to have to create some type of loops to regulate the order of the genes that are meant to be crossing over, because you still want to have the alleles of the gene properly aligned so you can make haploid copies of the chromosome that have no abnormalities to it. Let me show you this picture that's on the next slide. Hopefully that'll kind of clear it up. Okay, so let's go ahead and let's take a look. We're going to do two scenarios. The first scenario is when you have a pericentric inversion that's labeled A on the left-hand side. That is when it involves the centromere. And a paracentric inversion, obviously, is when it doesn't involve the centromere. Okay, so do me a favor, let's start in the paracentric example. Let's go all the way up, and what we see happening over here is that we're looking at inversion heterozygotes, because this person has an example where you have a normal chromosome, and then you have one that has the inversions, okay? So here's our normal order, and here's the inverted order. So this person is walking around, living his best life, no problems at all. But however, when it's time to start producing our gametes, we know that we have to undergo meiosis, and meiosis needs to do crossing over, where it's swapping the alleles from homologous chromosomes. Now, in order to do that, we're going to have to make sure that the alleles are properly aligned. So in my example right over here, I see that in the mutated version of my chromosome with my inversion, my order of the gene G, F, E, and D are opposite of what they're meant to be in the wild type. So what I see happening is during meiosis, when the homologous pairs line up, they're going to go ahead and create this thing called an inversion loop. And the inversion loop just basically means that the chromosome with the abnormality is going to have to loop itself in a very irregular pattern to make sure that the genes are properly lined up in the order that you're used to. So notice within the loop how you have now the restored order of D, E, F, and G. So the inversion loop is meant to line up the proper alleles within the homologous chromosomes. The problem with this is, though, is that when the inversion loop is formed, once you have your crossing over that occurs, most of the time you have a high amount of abnormal chromosomes that are produced at the end due to the abnormal shape of the inversion loop. So we can go ahead and come down here to the bottom of our products after meiosis, and what we see happening is that we have two cases of chromosomes where you have duplication and deletions that have occurred. So you have two of them that are okay, and you have two of them that are duplicated and deleted. In our other example, if we come over here to the paracentric inversion, what we see happening now is that the order of our E, D, C, and B has been altered. So once again, when the homologous chromosomes pair up, they're going to create their inversion loop. Oh my goodness, look at that. They will try to complete their crossing over, and at the end result, what we see happening is that now we get our haploid copies. And once again, we see that they are not completely normal. We have at least two of them that have abnormalities, including one that is acentric, meaning it has absolutely no centromere, and another one that is dicentric. And dicentric means that you have two centromeres. Now, this is not a permanent formation because when the chromosome goes into anaphase, it will break it off and then the chromosome will completely disintegrate. So what we see happening is that individuals that have inversion heterozygotes, they are unsuccessful at producing 100% viable gametes. They will have, as you can see from this example, about a 50% success rate. The other 50% of gametes will have abnormalities and most likely will cause semi-sterility to occur within the organism. Another abnormality that you can see within your chromosomes is translocation. So translocation is when we're going to start swapping information, genetic information between non-homologous chromosomes. And usually what we see happening is that this can be caused due to the fact that there might be a chromosomal breakage and the DNA repair is not done properly. The piece that's broken off is put onto a wrong chromosome by mistake. Or if you have crossing over that occurs between non-homologous chromosomes. So here's a look at what I was just chit-chatting to you about. So for instance, in our first example, we're going to take a look at some chromosomal breakage and improper DNA repair. You can see that we're looking at chromosome 22 and chromosome 2. And let's say for some type of reason, maybe like an environmental factor comes into place, and we see that there is a breakage that occurs. And when the DNA repair mechanism is activated, we see that a horrible mistake is made, and instead of returning the breakage end to its proper original chromosome, we see that they are swapped. Now, in this case, not only are you doing a translocation because you have swapped the location of genetic information on the chromosome, you're also doing a reciprocal translocation because both of the chromosomes have received additional information. The example on the right-hand side is when you have crossing over between non-homologous chromosomes. So as you can see right here, you have chromosome 1 and 7 that are lined up, and there is a mutation or a random event that occurs that allows the crossing over to occur. Now, since the crossing over has occurred in both, once again, it's an example of reciprocal translocation. Now, because the reciprocal transformation is equal on both ends, that means that we are not changing the total amount of DNA. So sometimes they also like to call it a balanced translocation. So if you come across that term, it's the same thing. Reciprocal and balanced translocations are usually good. Let's see. It says reciprocal translocations like inversions are usually without phenotypical consequences, and part of that is because you still maintain all the genes that you require. However, do keep in mind that just like when we were talking about inversions, we really have to take a look at the overall position as to where this is going. In a simple translocation, we see that it is an unbalanced translocation. And simple translocation, as we saw before when we started introducing these terms in Chapter 8, is when you only have the transfer of genetic material onto one chromosome. These unbalanced translocations tend to have a much higher degree of phenotypical abnormalities, and there have been lots of examples where they can even be lethal to the organism. All right, it says right over here, it says individuals carrying balanced translocation have a greater risk of producing gametes with unbalanced combinations of chromosomes. So this is very similar to when we were talking about our inversions, the way that when it's time for the gametes to form, that now you have to properly lining up before you have success in crossing over. And also what we see happening is that during meiosis 1, we see that the homologous chromosomes can synapse with each other. And if you want to have this done properly in a translocated chromosome, just like when we did our inversion, you're going to have to do a translocation cross. So there are additional steps that are required in order for you to properly align the genomic material and make sure that you produce a viable gametes. In fact, what we can do is we can go ahead and take a look at an illustration that can show you the 3 different ways that we can do the mitotic segregation. And in fact, before I show you the illustration, let me go ahead and show you the terminology. Okay, so remember, our goal here is to now take someone who has balanced translocation and go ahead through the process of meiosis and produce a viable gametes. But in order to do that, we're going to have to make sure that we properly align the alleles back to normal. So we see that we can have basically 3 ways of doing this. We have what we call alternate segregation, which is when the chromosomes on opposite sides of the translocation cross-segregate. You have adjacent segregation. The first option is to do adjacent non-homologous chromosomes that will segregate within the same cell. Or your second option is to do adjacent homologous chromosomes, which also segregate within the same cell. I will have you know that for the adjacent segregation, these are usually an unbalanced transfer, which will usually lead to inviable gametes that are being produced, meaning that sterility is very highly likely. The alternate segregation, when you have the chromosomes on the opposite side of the translocation, if this is successful, it tends to produce viable gametes that are balanced. So let me go ahead and put this in an illustrated example. So let's go ahead and click over to the next slide. And here we can see our translocation that's occurring. And it really looks like there is like a chromosome party that's happening over there. But unfortunately, what we've done is we've done lots of translocations. So now what we need to do is we need to go ahead and we need to start separating them so we can go from diploid to haploid. So over here on this side of the illustration, you see your 3 options. You can see your alternate segregation. And what we're doing over here is we're taking the chromosomes on opposite sides of the translocation cross so that they can segregate within the same cell. And when we do this, we tend to go ahead and be able to produce 2 normal haploid cells, which is fantastic, and 2 cells with balanced translocation. And because it's a balanced translocation, that means that the likelihood of having endophenotypical abnormalities is very low. So this is a very good outcome that we're looking at. And this is my attempt of making a smiley face. Our second way of separating our translocation chromosomes is to do the adjacent segregation. Adjacent 1 segregation is when we're going to go ahead and do non-homologous chromosomes and get them to segregate. When we do that, unfortunately, we end up with 4 haploid cells that are all unbalanced. And if we go ahead and do our adjacent second segregation, which is very rare, as you can see right over here, what we're doing in this case is that we're doing our adjacent homologous chromosomes to segregate. And that will give us 4 haploid cells that are also unbalanced. So both of these scenarios are not a favorable outcome and can cause, like I said to you, semi-sterility. So your option is really to have a high amount of alternate segregation so you can produce the most viable gametes possible. Now, what if instead of having just an issue with 1 chromosome, you have just a complete variation in chromosome number? And what I mean with that is we can have an issue where you can go ahead and just alter an individual chromosome or just your entire set of chromosomes. So this is what this part of the chapter, section 8.5, is going to start exploring. Aneuploidy is when you basically have 1 or 2 particular chromosomes that are abnormal in their number. Euploidy is when the entire set of chromosomes is varying in the overall numbers that you would expect in the wild type. And I'll give you lots of examples of these as we kind of start exploring them. But for now, just keep in mind that if you see aneuploidy, that usually means that there's only like 1 or 2 chromosomes within the set that are abnormal in their quantity, whereas euploidy usually means the entire set of chromosomes are not adhering to the wild type numeration. So for instance, right over here, so let's say we are looking at a fruit fly, and in the wild type, we see that it's a diploid organism, and we see that each each chromosome, as you can see, comes with 2 sets, right? Because it's diploid, 2N. Now, let's go ahead and go over to the left side, and let's take a look at an example of if you have your polyploid in the fruit flies, and this would be an example of euploidy, because what we see happening is instead of being diploid, the entire set of chromosome is not 2N, but 3N, so it's a triploid. Or how about this example right below it? This is where the entire set of chromosome, instead of being duplicated, is actually 4 times the amount. It's 4N. It's a tetraploid. So if you see that the entire set of chromosome is abnormal in its number, so instead of being diploid, everything's triploid or tetraploid, so instead of 2N, everything's either 3N or 4N, then we like to call that euploidy. So every single chromosome is affected because the numbers are off for everyone. On the other hand, if you come over to the right-hand side, we see aneuploidy. And aneuploidy just says that there's usually 1 or 2 chromosomes that are not behaving properly. So for instance, if we go back to our wild type, we see that we have 4 chromosomes, each of them come in a diploid set, and then now if we take a look at our mutations, we see that we have chromosome 2, that is a trisomy, not a diploid set because we have 3 of them instead of 2. So notice how that's indicated right here. Trisomy 2, because it affects chromosome 2, and that means that chromosome 2 has its regular diploid set, which we would expect, plus 1. On this example right here, go ahead and find what you're missing. Hopefully we all saw that chromosome 1 was missing a pairing. Notice how everybody else has 2, so that's fine. So in this case, what we would see is we have aneuploidy because it only affects 1 chromosome, and to be more specific, it would be monosomy. Monosomy 1 because it affects chromosome 1, and that's because the 2N that we would expect is actually missing 1. So whenever you see that the numbers are indicated with the +1, -1, +2, -3 next to the initial diploid, or if you look at a triploid or tetraploid model, you know that it only affects 1 chromosome within the entire composition. So euploidy is when all the numbers are off, the entire set is off, and aneuploidy is when it's just 1 or 2 chromosomes that are misbehaving within the set. All right, so just as I was mentioning to you before, aneuploidy is a variation in number of a particular chromosome within a set. Usually this can have abnormal phenotypes because it does cause an imbalance in the gene products. If an organism inherits 3 copies instead of 2, we say that the amount of gene product can obviously be increased to up to about 150%. This could be detrimental to the entire phenotypical expression, or it can just affect certain sections of the organism. It really all depends on which chromosome is affected by this mutation. So here, for instance, you can see with the normal individual, we have our 3 chromosomes that are diploid. If you have trisomy in 2, that means that you have an increase in your gene production. And if you have monosomy in 2, you notice that you're missing 1. So once again, your gene production is also affected. In this case, you're producing less than normal. Both scenarios could be detrimental to the state of the organism. I will let you know that a lot of times if there's abnormalities within the chromosome number, it will cause what we call spontaneous abortion, which means that when the two gametes merge and the fertilized egg, the zygote is formed. Whenever it notices that there's an abnormal amount of DNA or genomic material, the zygote will stop developing and will go ahead and induce apoptosis. So the cell will not continue developing on for further on. We do know that there are some exceptions to the rules because there are plenty of disease models that are out there in humans where you have individuals that are either missing or have an additional chromosome to their set. And probably the best example of aneuploidy that we see in clinical applications for humans is when we take a look at patients with Down syndrome. In Down syndrome, most likely the patient has trisomy on chromosome number 21, and it turns out that of all of the chromosomes that we have, chromosome 13, 18, and 21 are the ones that are most likely to be viable if they have a third copy. So you can go ahead and take a look at table 8.1, and you can see the frequency that it's been reported. And a lot of research has indicated that if the third set occurs anywhere else on the autosomes, that it induces the spontaneous abortion that I was mentioning to you beforehand. But for some reason or another, 13, 18, and 21 are more viable or more able to accommodate a third set. We do see, though, that the patients will develop some mental deficiency. Usually there tends to also be some abnormalities when we take a look at some physical contributions, some facial deformalities. So there are some phenotypical abnormalities that we see. Now, I also want to point out that in the previous chapters, we've also discussed things like Klinefelter's and Turner syndrome when we were talking about X inactivation, and that's because there has also been clinical applications that have shown that you can have an abnormal amount of sex chromosomes. So for instance, if you have a patient that is born with Klinefelter's, then what we see is that patient has 47 chromosomes because they have 3 sex chromosomes, not 2. In that case, they have an XXY. If a patient is born with Turner syndrome, they have 45 chromosomes instead of 46, and that's because they only have 1 sex chromosomes. So you can have aneuploidy occur in both your autosomal as well as in your sex chromosomes. And with the sex chromosomes, usually what we see is that it tends to affect the reproductive success of the individual, and it will also tend to hamper the fully developing of the sexual reproductive organs when the patient has time to go into puberty. This will obviously all depend on which version of abnormalities they have. You want to keep in mind that when it comes to Klinefelter's and triple X, usually the effects are very minimum because you have your X inactivation that will come into play that can downplay the excessive protein production from that extra X chromosome. All right, and then euploidy, as we were chit-chatting beforehand, is when you have a variation in the entire set of chromosome. So instead of being diploid, maybe you are looking at a triploid, so a 3N copy, or you're looking at a tetraploid, a 4N copy. Most of the time, the euploidy is not well tolerated. With animals specifically, it tends to be a lethal condition. You will have high rates of spontaneous abortion. We know that for humans in particular, 46 is our magic number. Any alterations to that, whether it's going to be an aneuploidy or a euploidy, tends to be detrimental to the system. We do see that there are some examples, and we've actually already discussed one in our previous chapter when we were talking about sex determination, and I had mentioned to you that we can take a look at haploid versus diploid when we take a look at bees. So let me go ahead and switch over to the next slide where I believe I have that posted for you again. Here we go. So some variations that we see happening is in our honeybees. We see that it's quite normal for them to be haplodiploid. And if you might recall, the males are going to be the end result of unfertilized eggs. So they are going to be the haploid copy, the monoploid, only one set of chromosomes. Whereas if you're taking a look at your females, they are the end result of fertilized eggs, so they are diploid. And it seems to be working perfect for that particular species. We also know some examples, like for instance, the little frog hyla that you see right over here. They are able to have some variation in their total number of chromosomes, and they seem to just create two separate species, but that are completely viable. Humans, however, do not have that luxury. What we do see is that even though we are not in an entire organism-wise are able to tolerate high or abnormal numbers of chromosomes, we do see that certain tissues that we have in our body have the tolerance to be polyploid. And probably the most impressive one is going to be the liver. There have been lots of studies shown that liver cells can go ahead and do this thing called endopolyploidy. Endopolyploidy. And endopolyploidy basically means that the liver cell has an extremely high tolerance to varying the amount of genomic information that the individual liver cells have. There have been cases where it's common that most of the liver cells are triploid, so 3N, or tetraploid, 4N, whereas the rest of the body is normally diploid, with the exception of the gametes, obviously, which are haploid. And there's even been some examples where the liver cells are octoploid. And scientists believe that the liver cells are tolerant to these number variations because it allows them to produce a massive amount of protein than they would normally be able to produce in their diploid condition. And the protein is so essential to the overall homeostasis and well-being of the entire body. So that's pretty cool. We also know that you have what we call polytene chromosomes. These are chromosomes that are very enlarged and very easy to study. Here we go. Here's an example of a polytene chromosome. This slide you would most likely come across, and you might even have seen it if you remember, in your general biology lab. It's one of those slides that we tend to kind of throw at all the students when we're discussing chromosomes for the first time. It is taken from the salivary glands of the drosophili of the fruit fly, and what we see happening is within the salivary glands, the chromosomes will continue to replicate within the cell without actually doing cellular division. So as the end result, what we see is we see a massive amount or a massively enlarged chromosome that makes it very easy to view under a light microscope. And the fact that we call it a polytene chromosome hints at the fact that as it's doubling in genomic information, it tends to bundle up. So you could see right over here, it tends to bundle up. And these bundles make these band-like formation that once again makes it very easy to visualize it under the chromosome, under the microscope, excuse me. It is an excellent slide to start studying the structures of chromosome. So here is your little polytene chromosome. I also definitely have to mention that even though animals on a lot of organisms have a very low tolerance for altering our chromosomes, we see that plants are very common to have polyploidy. And we see, as you can see from the numbers on the slide, it says about 30 to 35% of ferns and flowering plants are polyploidy. It turns out that it allows them to express at an amplified rate their phenotypical expression, like the size of the flowers, the amount of the flowers, also the fruits that they're able to produce. So the plant has more of a robust structure. It tends to be larger, which is fantastic for the organism. We do, however, run into an issue when it's time for replication purposes, because if you have an odd number or an abnormal number of chromosomes, remember, it makes it very difficult for you to kind of divide that number in half to produce viable normal gametes. So here, for instance, you can see that you have the polyploids having an odd number of chromosomes, which means that when it's time to separate, they cannot have an equal separation. So here you have a triploid organism. There is an unequal separation, which means that what we're having here is an issue of high sterility that comes into play. This usually means that the plant is not able to reproduce at any rate or at a very low rate. So in often cases, it can cause sterility. There's also been cases where it can cause seedless fruit to be produced because the genes, are not able to separate themselves equally. Alrighty, so I do believe that this is our last slide for Unit 1. Go ahead and take your time, peruse through the notes, and like I said to you before, I am available through email in the discussion session, Blackboard Collaborate, whatever you want to do. And as we're coming to the end of Unit 1, make sure that you prepare your notes because that means that our first exam is going to be close by. Make sure to reference the class schedule for all those due dates. And I do want to emphasize something that I had said all the way at the beginning of our Unit 1 lecture, which is that Exam 1 and 2 will be heavily based on our lecture material and our PowerPoint. So I would recommend that you peruse through the chapters in the book, maybe do the Learn Smart modules, the little smart book icons on Connect, but definitely make sure that you know your PowerPoints inside out. And if you're not sure about anything, go back into the book, read up more about it, send me that email. Don't be shy, and then that way we'll do great on our first exam. Alrighty, so this is the end of Unit 1, and the next time we'll meet, we'll start Unit 2.